So welcome everybody. I'm Gaëlle. I'm the technical manager of the International Brain Laboratory, and I'm here today to present a couple data sets that we acquired as part of our collaboration. So the first one is obviously our flagship data set called the Brainwide map. But as Buzz here would say, I would like us to go a bit beyond just this Brainwide map data set, and I'll showcase to you a few other data sets that are not yet available, but that hopefully will be really useful in building foundation model. So to give you a sense of timeline here, we're here at COSYNE, and our Brainwide map dataset has already been released in both ONE and WB formats. And in this talk, I want to expand to 4 new datasets that will be available either this year, hopefully, or next year. This is really not an exhaustive list. We have much more datasets coming in, so I'll just discuss this as bonus at the end. And if you want to use any of these datasets, please come talk to us. We are a bit slow in doing the release, but we really want these datasets to be used. All right. So I'll go through these datasets in chronological order here, starting with the Brainwide map. But going through these, what I really want you to get out of is what does each of these datasets enable you to explore, and when you keep looking at the new dataset, how each of these build up on the analysis that you could do with the Brainwide map. Okay. So here's a recap of the modalities that we acquire in the Brainwide map dataset. So we record in mice doing a behavioral task. The mice is presented with audio and visual stimuli. We record its body position through cameras, and we record the neural activity using 2 neuropixels probe at once. And I want to mention here the sampling frequency of 30 kilohertz, because you'll see that in another dataset, this is very different. And the mouse has to respond using a wheel. So the specific task is the following. The mouse is presented with a visual stimulus on the screen, on one side of the screen, and it has to turn the wheel to move it to the center, in which case it receives a reward, otherwise it's an error. And we vary the contrast of the visual stimulus from 100% down to 0, and we also vary the probability of the stimulus being on one side or the other of the screen in blocks of trials. And this enables us to build psychometric curves, which are typically shifted if you take one block of trial or the other. So this is the standard task of the IBL, and all of the datasets that I'll present today I acquired in expert mice in this task. However, we have bonus dataset whereby we, for example, record throughout the learning of this task and also in other task variants. So again, if you're interested, just come talk to us and we would be happy to collaborate. All right, so the Brainwide map dataset is a rather large dataset acquired in 12 labs. In total, we have nearly 700 probe insertions. In one experimental sessions, there were 2 neuropixels probe inserted in a mouse brain, and typically one is at a so-called repeated site that we acquired in each and every mouse. And if you concatenate this data together, this yielded 45,000 good units or so-called neurons. And if you look at the activity of these concatenated neurons, you start to see patterns emerging, such as, for example, a widely spread burst of activity just following the reward delivery at the time of feedback here. So I hope that you can appreciate that this data set should hopefully be useful in building foundation model, but just not because of its scale, but also because of its quality. So I just want to make a point here is that we were really thorough in vetting the quality of our data set. This is a paper that explains all of the metrics that we've developed, looking just at the data in the repeated site. And what you can hopefully appreciate is that we basically throw out nearly half of our data due to quality control constraints, okay? So this is the same kind of vetting that we went through for the Brainwide map, and we hope that this can become therefore a really valuable data set for the community. Okay, so now that we are happy with our data set and its quality, the main question is, what does it enable you to explore? So obviously, this Brainwide map dataset was designed so as to relate the activity of neurons, either like through local field potential, action potential, or the spikes that you can extract from it, and the behavior. So that can be points that you can track on the video data, continuous variables like the position, or discontinuous variable like the leaking behavior. So we've started to dig into this relationship between neural activity and behavior in a paper that you can see here, but there's really so much more to do. So we really encourage you to grasp this data set. I'll link to a tutorial at the end of the slide, and just expand. Please use it. I'll go through now 2 example analyses that we've done, just very briefly to give you a sense of what you can do. So here's one of the questions that we were asking. we would like to know what are the brain regions that significantly encode the position of the visual stimulus on the screen. So this is a rather simple question. And here you can do it using two different methods. One is just looking at single cell activity. Is it significantly modulated by the task variable at hand? And another is using the whole population and doing decoding and asking, can you predict the task variable? So for this particular example of the visual stimulus position, what was interesting is to see that only a handful of brain regions were actually significant, typically in visual cortex here. But when we looked at reward, this was much more widespread. All right. So this is the kind of analysis that hopefully is interesting to you. But in terms of building foundation model, I know there's so much more to do. So yeah, please reuse it. So this is the end talking about the Brainwide map. And now I would like us to turn towards new data sets. And I'll go in chronological order here, starting with what we call the EFIS Atlas. So if we take back the image of before, another thing that you can explore is the relationship between neural activity and brain region. So in other terms, are there specific electrophysiological signatures of this brain region that really characterize them? And this is something that we can see already by eye. So here, these are raw voltage traces in the action potential band of 3 different recordings spanning 3 different brain regions. These snippets are really small, like 40 milliseconds in time. And hopefully you can already appreciate that the activity is completely different, such as, for example, in cortex, it's really mellow compared to what happens in cerebellum. So here we can really see by eye, there are differences across brain region, and we want to characterize them. So the way we do this is that we extract from each electrode channel here, so each horizontal line in this plot, if you like, what we so-call features, such as, for example, the root mean square, or let's say the envelope of the action potential signal, or the power in the LF band, or some metrics that characterize the spike waveforms. And just to give you a sense of scale here, this tiny snippet that you see is 40 milliseconds, but we have 10 to the power 5 of such snippet in 1 hour recording, and we have nearly 1,000 recordings. So processing this is actually a massive challenge in terms of data infrastructure, and this is really led by Olivier Winter, who's our lead software developer at IB. Okay. So this is to give you a feel of what this looks like for a given recording. This is a recording spanning isocortex, hippocampus, and thalamus. Each of these columns is a so-called feature. And if you look here to begin with at the spike count in the thalamus, you can see it's slightly higher than the hippocampus, which also probably relates to the fact that the root mean square of the AP signal is also slightly higher because you have just more spikes probably. And on the contrary, you can see that in the hippocampus, the LFP power is much stronger than in the thalamus. So you can extract all of these features for all of our data set. And here, this EFIS Atlas data set is nearly double in size compared to our Brainwide map in terms of the number of insertions that we use. And that's because we care a bit less about the strict behavior criterion that we had in the Brainwide map. The mouse can perform a little bit less good, and it's still fine to use it in this particular data set. Okay. So then once we have extracted all of this, we can start to build lookup tools. So this is an example of a website where you can see the median root mean square of the LF signal, and hopefully you can appreciate that it's much stronger in cortex and hippocampus than in other brain regions. So there are clearly like relationships between these brain regions and these electrophysiological signature. So the first thing you probably want to do is build an AI agent to do decoding. So I know this is not a foundation model, this is just to give you an idea of what you can do. So given all of these features here, the question is, can you predict the brain region that the electrode went in? And you can. So here is an example of a random forest classifier built by Olivier Winter that outputs you cumulative probability for each of these electrode channel. A color here is a brain region. And if you take the argmax of that, you get your predicted label, and hopefully just by eye, you can see that this is not such a bad predictor, okay? But now going maybe towards foundation model, here's another example of using this electrophysiology atlas dataset. So this is work done in the Paninsky lab by Ryoo, and she was interested in just using spike sorted units feature, namely 2 features. One was the waveform and one was the autocorrelogram of these features. And she built a model using multimodal contrastive learning. And what was really beautiful to see is that the embedding formed by this model, who didn't know anything about brain region, actually really clustered into these known brain regions. For example, you can see this cluster of points here that are pink, which are a specific class in this embedding, really clusters nicely around the thalamus. Okay. All right, so to summarize this data set, what we hope to provide you with is a table of features, so you don't have to do this heavy computation yourself. Also a function to compute it on your own recording. We would love to see the Allen data set do the same, for example. And we are going to provide lookup tools such as websites so you can easily navigate these. I went through one example application, which is a brain region decoder, where there's so much more to do. So if you want, we can discuss after. And we are hopefully planning a release later this year. Okay, so now I'm going to look at another data set. And so we go back into our picture from before. Another thing that could be interesting is to look at intra-area dynamics, and typically within cortex. But in our Brainwide map, we capture maybe a few dozen cortical cells in one particular recording, and that's just because our probes are really elongated. And so now we want to design a study which is basically orthogonal, where we can capture many, many cells in cortex as one. And this is work that's led by Samuel Picard at UCL in the Carandini and Harris lab. So using mesoscope, which is, I will show you, but a big like 2P microscope, what you can do is tile nearly the whole dorsal cortex in layer 2-3. So Samuel here is using 3 different positions to place this really large 5-millimeter window in the brain. And what you can hopefully see is that in one of such windows, you can access multiple brain areas at once. So to give you a better feel of the experiment, here's the apparatus, this 2P random access mesoscope. And using that, you can really try to measure at a certain depth excitatory cells here in the brain. When you do the experiment, you have to select field of views, typically 6 or 8 for a given session. And within that, you get regions of interest, which are typically either the somas of cells or dendrites of cells, or just like weird blob that you have to disregard. So Sweet2P, which is a classification algorithm, rejects 50%. We are developing our own algorithm for classification. Happy to show you pictures at the end if you want. But just to give you a sense of scale, so in one field of view, we record 2,000 of such arrays, half of them are disregarded. We record 6 to 8 field of view for a given session, and that yields 5,000 to 7,000 good cells for one session. So this is really nearly 2 orders of magnitude higher than what we have in the Brainwide map. And this is where I wanted to mention sampling frequency, because that obviously becomes a trade-off, although it wouldn't make sense to record at a much higher rate because she can be slow in its nature. Okay. So that's just to give you a feel for what you get in just one session. Obviously, you get the animal's behavior. In one field of view, like we said before, you can get nearly 1,000 good cells, and you get multiple of these field of view in one session. And what's interesting, and we'll see this in the next slides, is that you can actually record from different brain areas at once. And if you take just one of the lines in this raster plot, you can really do similar analysis as before, where you see, for example, here a cell that is beautifully responding to visual contrast. Okay. So the reason why I was mentioning that recording from multiple regions is interesting is really for you in terms of building a foundation model. So in one experiment, you can record from different field of view, and one can be from region A and the other from region B. And what's cool with the mesoscope is that we can actually acquire the same field of view over days. So on the next day, you could record from region A and region C, and so on, and so on, and so on, all right? So for a foundation model, what could be really interesting to see is that probably you can predict the relationship between region A and B, and also between region A and C, because they've been acquired simultaneously. And the question would be, can it generalize this and predict the relationship between region B and C, which haven't seen each other physically? Okay. And just to show you that it works, here are example 3 cells tracked over many, many sessions. And so again, if we say in one field of view, you acquire roughly 1,000 good cells, you can track 70% of them across all days. Okay. So to show you where we are in terms of data acquisition, all of this has been acquired in 9 months in 82 sessions, totaling nearly 300,000 cells. That's really large. And so we hope for data release later this year. Okay, so now I'm going to go quickly into the 2 other last data set that should be hopefully available next year. So starting with one on multi-area. So if we start again by taking our picture from before, one thing that could be really cool to study is the information flow across areas, right? So we started doing this in the Brainwide map paper, and here's a picture on grandeur causality. But actually, this Brainwide map dataset was really not designed to study this, in the sense that, yes, we had these longitudinal probes that capture multiple brain regions, but a pair of brain region was never repeated, right? Because we were just trying to tell the whole brain. So this is where our work in the Steinmetz lab led by Kim Miller really comes in, and she's trying to record in targeted regions that are anatomically connected multiple times, 5 times namely. And so she's using typically 3 fork-shank neuropixels probe and tries to target very specific brain regions. And these brain regions are the following. There are 16 of them ranging from cortex to cerebellum. And the way that they were selected, so waiting for you to take your picture, is because either they had strong postsynaptic or presynaptic connections. So each of these, like blue square here, becomes what we call a targeted brain region pair. So there are 60 of them. And here's just a plot to show you how far we are in the recording. So we're hoping all of these to become dark green at some point, which means that they've been targeted at least 5 times. So in terms of number, they've recorded from 21 subjects across 110 sessions already. Half of these pairs have been recorded at least once, and a third have been completed. So we're hoping for a data release in 2026. Okay, and the last data set I want to mention is about behavioral modeling. So it's not so much on the neural data side, but the idea is to give really a rich context that is about behavior for you to do your foundation model. And the idea is that the behavior of the animal is much more complex and much richer than what can be captured with these variables such as wheel position or the movement of the paw. So one example of that is to ask whether the animal is engaged or disengaged in the task, right? So here is work continuing on a model that Zoe Ashwood in the Pilo Lab has developed, now overtaken by Alberto Pezzotta at UCL. And it's an HMMGL model that would give you 4 states, either 2 engaged states, so this can be seen by the sharpness of the psychometric curve, or 2 disengaged states where basically the animal just turns the wheel on one side regardless of what's happening, okay? And what's cool with this model is that you get a probability for each of these states to occur at a given trial. So you can see in the very first block of trial, the animal is quite engaged, but oscillates between being biased left or right. But as soon as the stimulus is really biased towards the left side, then the animal is engaged, but also biased towards the left side. So we think that this is the sort of data that you could really input in your model, and it could give a better context into what's going on. Okay, so just to recap, we've looked at 5 datasets throughout this talk. We have really so much more. So please contact us if you're interested. We have wide field imaging throughout the whole dorsal cortex. We use fiber photometry to record in specific neuromodulators. We have different tasks. We acquire also throughout learning, in disease models, in perturbations, you name it. So just come talk to us. We will be more than happy for this dataset to be used. So this obviously takes a village. So I cannot acknowledge enough everybody who's on these slides, and obviously thanks a lot to the workshop organizers for us to help us disseminate this data set, because we really want this to be used. And here, just if you want to take a picture, these are all of the papers that I mentioned in the talk. Happy to toggle back to the slide, but the slide that I really wanted to take a picture of is this one. 1, 2, 3. Which are pointers to basically first our mailing list. So if you want to use these data sets and are eager to wait for them to come online, just join here. If you have inquiries and want to use data sets that are not released, just contact us. And finally, here's a pointer to our tutorial on how to get started with a Brainwide map. So just go on our website under resource data and you will find it.